Okay, so it looks like a lot of people already joined us, and it's 7:05, so I'd say we can get started now. Hi everyone, welcome. My name is Chiara Campana and I'm part of INet-NYC City, which is hosting this event tonight. I will moderating tonight's discussion together with my colleague, Esther Wershof, who's also from INet-NYC City. And the title of the event tonight is Moving from Purely Experimental to Computational. And we have a great panel of speakers that will share with us the challenges and the rewards of using computational biology skills in both industry and the lab. So they're also here to answer any questions that you may have regarding this topic. So please, we invite you to drop any questions that you might have in the chat throughout the whole meeting at any point. You can direct your questions to either a speaker individually or the full panel, and then me and Esther will read those questions for you. So before getting started with our discussion and Q&A, I'd like to spend a few more minutes introducing what INet is and what we do at INet. So for those of you who don't know us yet, INet stands for International Networking and is an organization that's based in New York City. INet was founded in 2014 with the mission of providing support to international scientists in the STEM field. They're located and affiliated with institutions in the New York City metropolitan area. So we aim to offer professional development hub and opportunities, and we assist our members in building a community, a scientific community, and their own network. In particular, we organize a series of events throughout the years. These events range from career development to networking and social events, and we also promote and share research and work opportunities both within the US and outside the US. We also hold informative sessions on topics such as possible career pathway that are available both in the academic and private sectors. So INet is formed of board members like me and Esther. We're graduate students or scientists or postdoctoral fellow affiliated with research institutions in New York. And we also have a network of more than 900 research scientists, foreign scientists that are members of INet. And you can see on this slide names of many institutions that are part of INet. So if you're interested in becoming a member and start receiving our monthly newsletter, you can find out more about us on our website, which is INet-NYC.com. And you can also email us directly if you have any questions or if you have any suggestions on events that you would like to see in the future or any feedback that you might have. We also have open positions that are regularly posted on our website. So if you're interested in having a more active role and help us with events organization or our newsletter, you can apply for such positions on our website as well. Please follow us on our socials. We're very active on all major social networks, and we post our activities, events, and career tips. So before concluding this brief intro, I would like to, I have one more thing to say about our website. So on our website, we also post upcoming events. Our next event is already up on the website and you can register for it already. It will be held on Zoom on April 27, and we will have with us Christina Griever, who's from My Green Lab, and she will share with us tips on how to reduce the environmental impact of our research in the lab. So registration, as I said, is already open for this event, so make sure you register and you can find out more about this event and sign up for it just by following the QR code that's in this slide. So now this is the conclusion slides. I'd like to acknowledge our co-founders and all members on our advisory board, as well as our sponsors and partners. There are many institutions and organizations that throughout the years supported the organization of our events and continue to support us and collaborate with us. So thank you, and thank you all for your attention. I will now hand it over to my colleague Esther, who will introduce the speaker, and then we'll get started with this event.
Thanks, Chiara. Good evening, everyone. Thank you for joining us. We're going to dive straight in, and we're really fortunate to have four amazing speakers with us this evening. And I'm going to let them introduce themselves and talk a little bit about their journey through science and how computational biology has become part of their lives. First up, we can start with my friend Rabia. So over to you.
Can you guys hear me all right? Awesome. Well, first of all, thanks everyone for dialing in what I imagine is 7 o'clock, but you may be all over the world since we're global. And thanks the team for having me here. So my name is Rabia. I have probably have a bit of a weird career, so I'll walk through it very quickly. I will say two things. I'm always happy to chat. So if you want to catch up after, you can always ping me. Esther has my email address. And happy to talk through my career path one-on-one. So I have a PhD in mouse genetics, actually, a lot of spleen work and facts during my PhD a very long time ago, but I knew I wasn't going to be an academic. So I did an MBA at the same time down the street, and then ended up in the tech industry and worked at a company called Meta that built a biomedical knowledge graph. And I was responsible for building their commercial arm, pre-Series A and post-Series A. They've since been acquired by the Chan Zuckerberg BioHub, and you can use their product at CZI. And I was lucky enough then to get a postdoc at the Francis Crick Institute, where I met Esther, amazingly. I was working on iPS cells, so back on the bench. And they tell you frequently that once you leave academia, you can't go back. I'm a testament that that's not true, and you can do it if you really want to. But I didn't last very long. Which probably tells you something about me. And I ended up in a company called Benevolent AI, which is a pretty large computational drug discovery company, unicorn at the time and continues to be one. I joined when they were quite small and really fell back in love with the tech sector, looking at how we use machine learning to do target prioritization, run drug discovery programs working on glioblastoma organoid models, and then got recruited into a company called Sensine Health, which is a publicly listed company in Oxford, UK. And there I built their machine learning team. I took it from 0 to 50 when I left September last year, looking at how we use electronic patient record data, the equivalent in the New York ecosystem, which is probably really well known as Semaphore, and they're doing really great, exciting things. And then lastly, I left September last year to start my own company. We are actively hiring, shameless plug if you want to know more, both interns, computational, wet bench scientists, you name it. We were part of Y Combinator, which is an accelerator in San Francisco this year, and it's been quite a journey. We also have a special program dedicated to women that have taken time off and want to re-enter the workforce. So if you are or know anyone in that position, happy to talk about that as well. Thanks so much, Esther, and looking forward to the panel.
Thank you, Rabia. That's great. And, you've really inspired me, and, you've got so much get up and go and fearlessness in science. I think it's so easy to get kind of pigeonholed and think that, postdocing is the only option. Moving on, I've been to quite a lot of INet events, but I've never had a speaker on a train before. I'm going to hand over to Reyes to introduce himself.
Thank you. Can you guys hear me? I think my bandwidth is quite low, so, and I'm not sure if I'm frozen, because I think everyone else is frozen for me.
We hear you fine.
Okay, great, awesome. So I'll just assume you guys can hear me. So I'm Reyes. I'm traveling from Boston to New York City right now, so apologies for the thing, but you have a great view, potentially, hopefully behind me. Undergrad, I actually started off as a philosophy major. I actually studied my philosophy, actually wrote a paper on actually communism and Marxism and critical theory. And so now I run a you can see how people change over time. So yeah, after I finished my undergrad, I did a little bit of computational biology work. I did some internships during that period, and I got really interested in understanding, how we create new drugs and how we create new medicines. So at Sinai, and there I did work under Dr. Abner Schlesinger, where I studied and trained and learned about kind of kinase drug discovery. It's a particular family of proteins that are really interesting, and I thought they were pretty cool. And that interest and a lot of the work that I did, me and my PhD advisor sort of came together one day and was like, yeah, we can easily translate a lot of this work into potentially a company to something that can actually create new therapeutics. And I thought that was really interesting and very cool. And additionally, we had like a lot of support from other people. So one day I just applied for a few startup accelerator programs, and we got accepted into one called Petri, which is out of Boston. Yeah, similar to Y Combinator, it kind of accelerates you, gives you kind of initial seed capital, about $250,000. And we use that to start IChemy or AIChemy. We don't know how to pronounce the name just yet. And the goal of AIChemy is to integrate and to create what we call precision AI, or precision machine learning tools, to understand and characterize protein kinases and how do we optimally design new inhibitors for kinase drug discovery. So I finished my PhD in 2019, and then we started IChemy about 2 months later, and we got funding, and we really formally started in February of 2020, and then it's been about a year now, just kind of working full time at it, and yeah, happy to kind of share what we're working on. That transition from kind of studying philosophy into doing computational chemistry.
Okay, thanks, Reyes. I think it might be best if you turn your video off just to increase bandwidth, but thank you for that. Okay, so next up we have Sunny, who has very kindly agreed to step in last minute as a replacement for Milind, who unfortunately can't be with us this evening. But Sunny's background, as you're about to hear, is really relevant, and I'm really excited to hear what she has to say.
Thanks, Esther. Thank you for having me here. And so I probably have the most relevant experience. And also I just graduated from PhD for 2 years now, and I've been working as a computational scientist. So I'm really glad to be here and share my experience. So I started with also a pure biologist. In my undergrad, I did mostly research around human genetics. That was also when I started to have a little exposure to computational biology. And then I decided I want to learn a little bit more of bioinformatics. So I did a master's at NYU in bioinformatics program. That's also in a wet lab where the lab is just going from microarrays to whole genome sequencing. So that's also a great opportunity for myself to really gain some bioinformatics knowledge, and meanwhile also generating data and doing a lot of experiments. And then I decided to, at that point, I still couldn't decide whether I want to go for purely computational work or just be a bench scientist. So I did my PhD at NYU School of Medicine in a more molecular biology lab, but I still don't want to give up on my computational skills. So meanwhile, I did some short computational project in collaboration with some dry lab PIs, and also was part of the computational biology program. Then before the year I graduated, I did an internship at Biogen and in their AI department. That was my first experience really in the industry, and had a chance to see how things were applied in the real world, and how drug development process has been using really the analysis from developed from the AI folks. Then a year later, I finally decided I want to go for computational biologist. And more specifically, I was looking for opportunities in early startups because I think there are more chance to learn multiple skills in terms of both developing AI platform and also applying it. So that's how I end up being at Cellarity. We're a company that really trying to leverage single cell data and other genomics data to facilitate the drug development. And I've been mainly working on developing the AI platform, but I also had a great opportunity to really apply what I developed in real drug development programs. So I'm also in charge of doing data analysis for a couple of IO program and also hematology programs, and really sit together with the wet lab biologists, going through the data analysis and providing insights and guidance for the wet lab experience. So I think really having experience knowing how to do wet lab work really helped me for my communication and daily work in my current job. And I'm happy to share more later in the panel.
Thank you, Sunny. I think it's a great example of how you don't need to have, been doing computer science undergrad and, be really hardcore, deep learning, all this stuff to actually do impactful work in that in computational science. So lastly, but not least, we have Raphael, who is a little earlier on in his journey, but that's kind of exciting to see how he's picking up the computational skills at the same time. So please introduce yourself.
Thank you. Well, first of all, I'd like to thank you so much for the invitation. It's a great, it's the train. It's not the same train, but it's a train. But it's a great pleasure to me share some of my experiences, part of my pathway with you guys. And also, I would like to thank you to my colleagues that did the introductions before me, because they facilitate a little bit my life. So now I know where I can focus my talk, my introduction. I'm Brazilian. Most of my train was in Brazil. So master, PhD, the undergraduation, everything, most of these things all in Brazil. But I think that I'm the oldest in the group. I started the undergrad in 2003, third, right? And I had my bachelor degree in 2007, my first publication in 2006, and so on and so forth, PhD in 2015. And after the PhD, a couple of different postdocs, different places, different subjects, but always working with cardiovascular diseases, in a special two big fields. First of them, and the most important in the whole path, study of stem cells as a tool for the development of cell therapy for regeneration of the myocardial infarction, heart failure situations, very complex diseases, very complex to treat, and that requires regenerative approaches. So this is one of the big side, or maybe my main background is based on, because of the cell biology, stem cell biology, molecular biology, especially cardiovascular physiology. So in the middle of this mess, I also could be part of a very important project in Brazil, and because of the type of the project that I developed, I was trained to work with echocardiography. So I'm not a doctor, I'm a biologist, right? But I can perform echocardiography, analyze echocardiography. And during a year, 2013 to 14, I was invited to participate in a project in Brazil, in São Paulo, in the Heart Institute, specifically in the Center of Heart Transplantation, to perform echocardiographies in donors. So dead people with potential to donate their hearts, trying to optimize the quality and the numbers of the transplantations in our country. In that year, because of this project, we could increase in 30% the number of good, successful transplants in that year, and saving money, saving lives. And so this was one of my first real contact with patients, with people, with families. So apply science in a real life, real world. But okay, great. And after this big experience, this nice, relevant experience in 2016, I decided to try something different. So I moved to New York City to work with Dr. Sobi. And I met
Sorry
Chiara.
to interrupt you. I'm just keen to get onto the questions. So if you're able to just kind of wrap up in the next minute or so, that would be
Of course, perfect. And so at this point, I started to work with some computational stuff. Computational stuff for me was more like a tool to help me at some point with bioinformatics in the very first moment. But after that, and working in here in Sinai, to understand better what kind of things we can do or we can have with these beautiful tools that we can work to, for example, to predict better how a cell will respond, for example, to a drug, how a patient can, a population of patients can respond to a different treatment, thing like that. So anyway, I'm just starting these things, but I'm using a lot and as much as I can and diving as much as I can in this new knowledge, because I think that this can be applied for many other stuff. So in the end, when during the QRs, we can discuss more, but the point is, for me, it was like something very different, right? Just in the end, the last maybe 4 or 5 years, I'm trying to learn the things that my colleagues probably know a lot. On the other hand, I have a very huge background and skill in cellular molecular physiology, like that. So
thank you for that. I'm going to cut you off there just so we can get on with the questions. And I just want to say, we have some questions prepared, but we'd really like to hear from the audience what questions you have. So please put them in the chat. They can be whatever you like. They can also be related to, even if you're not really thinking about outside of academia, if you're doing a project at the moment where you're interested in getting quite specific, asking about how you can integrate computational biology, this is the place as well. So any questions you have, please put them in the chat and we'll try and get through as many as possible. But just to start, my first question is, in your work, how are you practically using computational biology and what's that interface with wet lab and dry? So I don't know if any of you want to come in on that. If not, perhaps we can start with Sunny.
Sure. Well, my work would be more straightforward, as part of my work is really trying to analyze the data that's generated from the wet lab, right? So we are generating a lot of, for example, single-cell RNA-seq data and also other types of genomics data. And we wanted to understand, for instance, how cells are respond to drugs. And by that, what you immediately do is obviously some single-cell RNA-seq analysis to really understand the gene profile changes and how is that change related to the mechanism of the drugs, and really to help us understand the disease aspect of the biology. And my daily work is very heavily associated with both the technology development, which is also a big part of the company, that they're trying to develop an optimized method to run high throughput single cell sequencing and other type of sequencing technologies. And another part is really the biologists are developing different assays and also trying to optimize these models that can better capture the clinical translation and the link between the preclinical models to clinical data. So that's also some aspects of analysis that we really need to gather from the clinical data and trying to link the molecular signature to what we observe in our preclinical models.
Yeah, it really sounds like a Cellarity. It's like very fluid, like you have that whole spectrum of wet to dry.
Right.
Reyes, do you have anything to kind of add to that on kind of the integration of wet and dry and how you're sort of practically using computational biology? Reyes,
Frozen.
can you unmute yourself?
Can you hear me?
Yeah.
Okay. So one of the biggest opportunities in kinases is to go after a set of kinases called the dark kinome. And these are kinases that are fairly understudied, and we don't have a lot of information. So one of the biggest partnerships that we've signed focuses on creating biochemical tools to probe dark kinases, and do that in a very scalable, high throughput way. So this is a really exciting opportunity for us, where we're working hand in hand with one of the biggest assay development companies, a huge data set to understand the physiology and how these dark kinases work, and then see if we can use any of these dark kinases as potential targets for drug discovery. And then we take that data and we use a lot of bioinformatics algorithms, we use a lot of analyses and target prioritization algorithms to really filter and determine which targets to really focus on. And then the kind of second half of that equation is the computational chemistry that we do in terms of identifying lead compounds and finding novel inhibitors for these targets. And so that's a lot of the work that I did kind of last year, building out the pipeline and the model, which is a lot of cheminformatics. How do we use AI and generative algorithms to design completely new compounds that have never been seen before and have predictable pockets and predictable features. And so that's really kind of at the intersection of what we're trying to do, is combine wet lab and our knowledge from that with computational chemistry to really push forward new and novel medications.
That's great. Thank you, Reyes. I guess just we'll put the as a you're doing really exciting things. We'll share all of your LinkedIn and company details, and anyone who wants to get in touch and find out more can do so. So there's a really nice question that I think kind of naturally follows on from that in the chat, which is that often industry jobs are you develop tools as part of a larger team, but in academia, you typically do things much more solo. And the question is, do you have any tips on how to actually build those kind of team working skills? So maybe we can come to Rabia first.
Yeah, I actually have a favor to ask everyone. If you could, like, in the chat, put your background, just so we can talk a little bit more to the audience and be useful with what we say for whoever is comfortable with that. Sorry, Esther, to throw a wedge, but that just helps.
That's a great idea.
My comments. I think that's a really great question. So the one thing I will say is that make sure you're targeting your applications for the right positions, right? So startups like ours have tons of internship positions. So if you're open to internships or fellowships, so we're going to have a fellowship program where you can do that while you are still working, will allow you to get that experience on your CV. But at the same time, there's a lot of roles now that are opening up, which are product owners and product managers, where you don't necessarily need to have these skills in terms of knowing how to do this in a larger team, but actually you can take the intersection of both worlds. And I can talk about that for anyone who's interested. But genuinely, the one-line answer to the question is either apply for internships or show that you've worked in a collaborative project, or try and run a Kaggle competition or something like that as well, to show that you can build end-to-end pipelines. And like I said, happy to talk about that separately as well.
Just a compliment. Even in the academia, you can have these skills probably, obviously, as I can see in the chat, with more publications. And obviously, how can you do publications? Collaborating, right? Not only in your group, but with peers from different groups, with similar or sometimes not similar, completely different fields of study, subjects, whatever. You can learn with your peers. And this definitely counts as an important experience to all of this, right? So collaboration. I many times here in the US, I saw and see people that really are working solo all the time. They have no ability maybe to look on the side and see people. So if you want to improve your abilities, not only the ones, the traditional scientific ones, but also your skills to talk with people, to interact, you need to look for the other ones, talk to the other ones, not only work solo, right? So this is kind of way that you need to change your way to behave in the lab, right?
Yeah, I think it's a great point.
Sorry, one more. I was just going through hiring some interns recently, and one of the things that I noticed from a lot of the applications is, also kind of gear your resume CVs towards like the thing that you're applying for. A lot of times, when I was asking for computational chemists, a lot of times I'd get applications that wouldn't really emphasize the computational chemistry, like a lot of the drug development stuff that they've done, or the cheminformatics stuff, or the tools that they used. And so it's really helpful to tailor kind of CVs and resumes towards the thing that you're applying for. So.
Yeah, absolutely. It's a great point. So just one of the things we've done is in advance, we sent each of our speakers a specific question that we kind of like them to answer, because we think they're specifically suited to answer it. So I'm going to come to Sunny first. So our question for you is, what skills and research areas do you think will be important for experimental biologists to learn or brush up on in the next couple of years to bridge that gap into kind of computation?
Okay, so I think one of the most important thing, and also we often look for, is experience and like real world experience on applying the computational skills to projects. And in grad school, I think there's really great opportunities, because right now for a lot of wet lab is generating high throughput data. So a great opportunity would be, if you're the person generating the data, you can also try to learn how to analyze the data. Instead of going to a collaborator, which is a pure dry lab person, you can really start by going to the website. There are so many open source tools and packages out there. Just try to learn from the basics, that how to really just start from like simple programming, trying to get this data analyzed, following some tutorials. There are like so many out there, and everybody is really open to help you if you really need like suggestions and have questions. I'm sure like there are computational biologists in your institute or like next door would very willing to help you. And I think that's one of the most important experience that I had in grad school. Really helped me to get at least into this job I currently have. And in addition to that, of course, if you want to go for more machine learning or like deep learning type of positions, it'll be also nice to really take some like serious courses and really to improve your skills and related knowledge.
Great, thank you. So now I'm going to ask the specific question to Rabia's specific question, but actually it was a question in the chat that I think will really nicely relate into it. So maybe she can answer that at the same time. So our specific question for you is, coming from an experimental background, how much computational biology do you actually need to know in order to manage people? And I know you manage many people who are, hardcore computational people. And then the question from Kyle is, he's a computer science student, and he said, do you kind of wish you'd done that instead? Would it have been more useful to know the computational stuff, like as compared to the biochem stuff?
So Esther, you're gonna hate me, but I'm gonna like turn this question on its head, primarily because I think, when I was doing my bench work, I always looked at like the RNA-seq stuff, and I was like, oh man, I wish I knew how to do that. And then when I got comfortable with that, I got introduced to this world of machine learning, and I was like, oh man, I wish I knew how to do that. And so there's always something else. And so actually, I don't code, but I've managed a team of over 50 machine learning people, and the company we're growing right now is also machine learning. And so I would say the world now is at a point where you can always train in something between Coursera, all of these different things. You can prove on your CV with Kaggle competitions that you're capable of delivering. And so what we look for, and when I'm managing people, I'm not actually looking like the basic things everyone can check off. You can say, I did X Coursera courses, here's my Git repo, here's proof I can do something. To Rayees's point, like when we ask for cheminformaticians, it's great if they have a PhD in cheminformatics, but if they had a PhD in medicinal chemistry, but have a massive Coursera Git repo profile, that is good enough. And what you actually need is people that care about what they are doing. And I know this is like a soft answer, but I don't think moving forward, any of these companies, whether it's Benevolent AI, whether it's our company, perhaps Reyes, I don't know if I can speak for you, but we are looking for people that can understand both worlds and speak both languages, and they don't necessarily have to be heavily trained in one. They have to understand both. So the roles that I've found the hardest to hire for are people that can have a deep domain understanding in one and can understand the other as well. So that is people with PhDs in biology but understand machine learning and can be the translation and the machine that speaks both languages. That is actually the role that is hardest to fill. When we had a machine learning application open, I had 300 applications in 5 days. It's no longer about having a CS degree, it's about delivering something and finding something you really care about. Sorry, I know that wasn't the answer you were looking for, Esther.
Just to add,
wonderful.
You called me out. So I completely agree. Like one of the best hires we ever made, he, exactly as you said, it's that crosstalk between multidisciplinary perspectives. So one of the best hires we ever had was a medicinal chemist, trained 20 years, patents in drug discovery, very traditional. And then what really stood out was that he had Jupyter notebooks where he was just playing around with, cheminformatics libraries and tools, and he showed us what we were doing. And it, that really spoke more than all of his publications. The fact that we can come to him and ask him technical questions about the machine learning, about what we're doing, but also come back to him because of his deep domain expertise in medicinal chemistry.
That's great, thank you. So I'm just going to hand back to Chiara now, who's going to take over the Q&A.
Okay, great. So in the meantime, we'll launch a poll. I don't know if everybody has seen the results. So it seems like the majority of the people in the audience had an experimental background, and only 13%. Not everybody has replied, but among the responders, only 13% have also a computational background. So I think some of the questions in the chat were not directly answered, but they were answered as part of other questions. I think one that I can see that hasn't been answered yet is if any of you on the panel has any program or conference that you'd recommend for networking in this computational field. If Rabia, you want to go first?
Yeah, I would say, so Ray, sorry to cut you off. Maybe I'll steal your answer. And so I would say don't wait for conferences. We are now in a remote world. The number of people that ping me on LinkedIn, I always make an effort to reply if you tell me why you've pinged me. Don't randomly adding people probably doesn't work. But if you reach out to someone and say, I really like what you're doing, do you have 10 minutes to chat? It's just 10 minutes on a Zoom call, 15 minutes on a Zoom call. So why would you wait for a conference? Why wouldn't you find these amazing companies that are popping up? I know Cellarity has gotten tremendous press with amazing, your CEO is incredible, the CDO is incredible. Reach out to these people now. I wouldn't wait for conferences. You can go to NeurIPS, you can go to IEEE and AACR, but I just go on LinkedIn.
Mm-hmm. Agree.
Yeah, I was just going to agree with that too. I also was just going to say that conferences like ACS, if you're a computational chemist, I think those are really good opportunities to go and network with people, not necessarily for jobs. I think the best way to get a job is, or opportunities is just by generally talking to people who work in your domain and then kind of building a rapport as well. That's always a really wonderful get in the door approach as well.
Okay, yeah, so if nobody else wants to chime in on this, I actually have one question. I don't think there's anything new in the chat, so I'll ask my question and then I'll wait for more questions to come in the chat. So I was wondering, I think Sunny mentioned before that she gets data from experimental lab. I think that was related within the company, but I was wondering if in your experience as a computational startup or as a computational company, if you collaborate with any university or any hospital, or where do you get the data from if you collaborate with healthcare organizations or any universities that provides data?
Yeah, definitely we do. We were trying to also get data just like, so we have our own sequencing facility, and which will process the data systematically and really generate in a format that that fits into the machine learning team. But we definitely collaborate with hospitals and also some organizations just to get patient samples, and also their like more large scale data sets that fit in different purposes, different drug design programs. And we also have like license, for example, digital libraries, drug libraries, which have like billions of compound structures, which that the chem informatics team really leverage to help really optimize the NCEs and drug design.
Great, thank you. Anyone else wants to comment on that? Okay, so I can see another questions. I think it was related to that if you collaborate with universities. And another question is if your company that you work for or your own company offers internship. And I think Rabia had mentioned that they are hiring and they have internships. So maybe you can elaborate on the requirements of those internships.
The full-time roles are online. I will say though, there's a ton of roles that are not online, specifically around product and ops. So you know, I noticed there's a whole bunch of web bench scientists. There's like tremendous opportunity in product operations and project management for people with that background. It's not like, I wish someone had told me that when I was graduating. No one did. I don't know why. And so we're hiring across those three roles. We have a fellows program, which is a number of hours a week where you basically work with us very closely on a therapeutic area that's of interest to you. And then we have full-time internships as well. Esther, happy to share my email with anyone that's interested to get more details, because the programs, actually, we just haven't gotten around to updating our website.
Sure, and actually just after this event, we'll be sending around, you know, with all your details, like LinkedIn page and everything, so people can contact you through that too.
Yeah,
thank you.
There is a more specific question about visas. I don't know if you have any particular requirements on J1 versus H1B visa holders.
I'm not a lawyer. I can ask my lawyer. I don't know. Sunny might have a better answer, or Reyes. Like, I've never worked in the US. We are hiring in the US, but we can deal with that on a one-on-one.
Okay, great. And another question is more general, is where do you see the major opportunities for computational scientists within the life science sector? That's open to anyone in the panel, I think.
Maybe a couple of years ago, I would say pharma, but now the things are changing dramatically, right? We can see many opportunities only in this panel, right? My example specifically, and considering the company that we are working with, cell therapy, there are many opportunities or many possibilities to us to implement mathematical modeling to try to predict potential issues and goods in terms of cell therapy. Imagine, for example, that we want to regenerate a cardiac tissue, a heart, injecting cells generated from stem cells, from iPSCs, for example. We can generate cardiomyocytes from iPSCs and inject them in the heart. Nowadays, one of the big challenges in this kind of approach is because when we are injecting the cells, they are not mature enough, and they are connecting to the host tissue and generating many arrhythmias, many contributing to other kind of problems that are hard to predict. Mathematical models, for example, that resemble physiology, 2D, 3D models that resemble electrophysiology, for example, of the heart, are models that can be very useful, including to a company that is working to try to generate a therapy that we want to be applied in patients. Why? Because we can predict potential pitfalls based on the cell type that we are using, maturation of the cells, and all these kind of things can be at some point simulated using computational stuff. So this is just one example to show that more than pharma, nowadays we have many other opportunities. Currently, I think that the fields are more and more open to computational applications in different, the point of the dry and wet, you know, for example, in my example, my office is a desk and a microscope. So I'm programming in my desk and running experiments in the microscope. So everything in the same day at the same time. And I think that the balance is important. So now more and more new scientists need to be aware of all of these things as more balanced as possible, right?
Okay, thanks so much, Rafael. We have many more questions in the chat. I guess everybody posted the question all together. I actually wanted to ask Reyes a question that I think incorporates a few of the last few comments that were made in the chat. I wanted to ask you if you could give us a general or an example about your own career about applying machine learning in drug discovery and pharmacological sciences, and maybe you can comment on the importance of knowing statistics for computational biology, and in general, how would you go to apply these specific computational skills in drug development?
Sure thing. Thank you for the question. Yeah, I mean, so my own kind of personal experience and with the company, you know, the machine learning happens at kind of different stages, right? So when you think of creating a drug, there's preclinical drug discovery, and then there's kind of a clinical component as well. And even within that, there's target identification, lead optimization. And so in my own experience, I developed algorithms for target identification, as I mentioned before, like deconvoluting dark kinome, seeing which ones are really important drug targets for disease. And then we used machine learning, particularly generative machine learning algorithms, to create really new chemical structures that we went to CRO Enamine to synthesize and to test in vitro. And a lot of them worked. And it's really exciting when, you know, things come together and like pieces fall into place. And then in terms of statistics, I think statistics is really important for computational biology. A lot of the analyses that you'll do involve making and having a deep knowledge of like which types of statistical tests you need to run, what kind of hypotheses can you ask, like what's your null hypotheses, and comparing kind of results. And so having kind of a good grounding and knowing how to apply certain approaches is, I think, critical for a successful kind of role in a statistics by a computational biology company.
Great, thanks, Reyes. So there is a question from Taliana. Being a data scientist seems like a popular job title now. Do you have advice on what companies are looking for to be a data scientist? I think perhaps this was answered somehow through some of the questions, but if anyone wants to add anything, feel free. Okay, I can go to the next one. More specific about specific languages that one should learn to join computational biology, Python, C, R, what languages you use, and if you have suggestions on which is maybe the most approachable language from someone that comes from experimental background to start and getting started in computational biology.
You're probably going to start a war with this question.
I think it depends. I think there's, for comp bio, there's a lot more in R, and then for ML, there's a lot more in Python. Remember, I'm not a computational scientist, so like take this with a grain of salt, and Reyes can probably answer better. But genuinely, from the people that I've worked with, if you can code in one, you can code in the other. So I think just pick one and learn it.
Yeah, I agree. I think if you maintain expertise in one of them, it's kind of easy to transition to the other. I personally started with R, and R has this package called Reticulate, which lets you run Python code within R scripts. And so it's a way of interoperating between two different languages, and you kind of gradually learn each. So I think the most important thing is just having a project, and learning a language while doing the project kind of gives you the best net gains.
Great, thank you. There's a question from Neda asking, how is the work situation in a startup company compared to big corporations? If any of the panelists wants to.
I think that's one of the most interesting questions. If you don't mind, Chiara, I can chime in. I can tell you why. When we interview, one of the questions I ask point blank is, why do you want to work at a 4-person company versus a 10-person company versus like a 1,000-person company? They're fundamentally different career trajectories. I know Sunny commented on this earlier. And if you are a person that likes structure and you want to have very defined roles and a very defined career progression, and you want to know what you're doing and why you're doing it, and don't like context switching or chaos or like Slack messages randomly 10 times a day, my suggestion would be probably don't work at a very early stage startup unless that early stage startup is like heavily organized. I've worked at three early stage startups. I have not seen one that's heavily organized, but that could just be my bias. Whereas if you're interested in a laser like upward trajectory in your career, step function promotions, and like massive learning at a very high cost of like, you know, just constantly iterating what you know and pushing yourself, then startups are probably a better place for you. And then there's everything in the middle. But know yourself before you apply to jobs. Personally, I ask point blank in interviews, and if I do it, I imagine other companies do it as well.
I need to agree with you. You need to know exactly how you work, not before the company, right? Because, yeah, for example, our startup is like this. It's from the zero to the sky in a second. Some things are crazy in terms of work, challenges, and also challenges related to funding, to money, to investors. Sometimes the company is perfect, sometimes it's like you need money and the money is almost in the end and the taps are all not really helping you. So yeah, if you are not very self-tempered, let's say, maybe you need to think better before try something like that. On the other hand, if you like challenges, usually a startup is a nice place to try, right? As you told, you can have a flash and from this you can be like there, up high, super fast, with massive learning, with massive pleasure in the end. But you need to know yourself. You need to understand how this works for you first.
Thanks, Rafa. And Sunny, I know before when you introduced yourself, you mentioned about the difference in startup. You get to learn a lot of skills. Do you have anything to add to these questions? Any tip for anyone?
Yeah, I completely agree with Rabia, and I think like the main reason I chose startup is because I don't like to always do one type of work. I really wanted to learn multiple skills, and in a startup, I think your impact is higher as a person, because essentially you could, like, I'm hired as computational biologist, but I'm also doing stuff with like machine learning scientist and also like program management stuff. So you learn a lot of different skills. You're not only responsible for a very small subset of work, but you feel like you're interacting with different people, learning a lot of skills. And also your job responsibility changes over time, because like you as the company grows, there are like more opportunities gonna come up, and potentially you can also shift around. It's more dynamic. So you will as time goes, you also learn what you actually wanted to do, what type of job interests you more. So there's also more opportunity that you can actually shift your career trajectory in the small startups.
That's great. So we're going to wrap up now. It's 8 o'clock and I'm sure you've all got better places to be. But thank you so much to our speakers and to all of you for your questions. We're going to be sending a survey out after this, and it's just going to have all the details of all the speakers who have made it very clear that they're happy to have further chats. We'll also be putting a blog up about the event, and this chat will be online shortly. So thanks very much for joining us, and have a great evening.
Thank you, everyone.
Thank you, guys.
Thank you, guys.